Have you ever wondered how your body juggles so many things at once? I mean, building DNA, getting rid of toxic waste, and producing energy all at the same time. Well, today, we're diving into two tiny but absolutely mighty amino acids, aspartic and asparagine, that are right at the heart of all this action. We're going to see how they're not just simple building blocks, but major players in everything from regulating your metabolism to even some really cutting-edge cancer treatments.
So, let's just jump right in with the question that really gets to the core of why these molecules are so important. See, they aren't just hanging around waiting to be put into a protein. Nope, they are these sophisticated couriers, zipping nitrogen around your body for some of the most critical jobs imaginable, like detoxifying ammonia in the urea cycle and building the very purines and pyrimidines that make up your DNA. They are truly the unsung heroes of our metabolic world.
All right, let's get a proper introduction to our main characters. Meet aspartate and asparagine. Now, you'll hear them called non-essential amino acids. All that means is our bodies can actually make them, so we don't have to get them from our diet. But don't let that name fool you for a second. They are absolutely essential for keeping our whole metabolic machine running smoothly.
Okay, this slide perfectly sums up their relationship. The best way to think about it is like this. Aspartate is the generous donor. It's the one that directly hands over nitrogen atoms to build other really important molecules. Then you have asparagine, which is made right from aspartate. Asparagine is the secure carrier, kind of like a specialized delivery truck, safely transporting and storing that nitrogen until it's needed somewhere else.
So, let's kick things off with our first character, aspartate. We're going to look at exactly how it's made and what its primary job is in the body, which, as we just said, is all about being that generous nitrogen donor.
The way it's made is just beautifully efficient. It's a single reversible reaction called transamination. You take oxaloacetate, which is a key molecule from the citric acid cycle, our body's main energy engine, and an enzyme called aspartate transaminase, or AST, just plucks an amino group from another amino acid, glutamate, and sticks it right onto oxaloacetate, and bam, just like that, you've got aspartate.
And this process, transamination, is so crucial. Think of it like a nitrogen swapping service inside the cell. It lets the body shuffle these amino groups between different molecules without ever letting free ammonia float around, which is super toxic. The whole operation relies on a helper molecule, a coenzyme, that's made from vitamin B6.
And here you can see just how important aspartate's donations really are. It gives one of the two nitrogen atoms needed to make urea, which helps us get rid of toxic ammonia. It also provides key atoms for building both purines and pyrimidines, the A's, T's, C's, and G's of our DNA and RNA. Plus, because it can be turned right back into oxaloacetate, it's a huge player in making new glucose when our energy stores run low. What a multitasker, right?
All right, now let's switch gears and focus on our second character, asparagine. Remember, if aspartate is the donor, asparagine is that specialized vehicle, perfectly designed to safely carry nitrogen around the body.
Making asparagine is a little more involved. So, we start with our donor, aspartate. Then, using some energy from ATP, an enzyme called asparagine synthetases adds a second nitrogen group. And here's the key part. That nitrogen comes from another amino acid, glutamine. It doesn't come from that toxic-free ammonia. This whole process is basically packaging nitrogen into a stable, non-toxic carrier molecule ready for transport.
And breaking it down again is just as simple. When a cell needs that nitrogen that's been stored, an enzyme called asparaginase just adds a water molecule to split asparagine right back into aspartate and ammonia. The cargo gets released exactly where it's needed.
Okay, so let's connect all this biochemistry to the real world, to the clinic. We're about to see how these metabolic pathways aren't just diagrams in a textbook. They have direct, sometimes life-saving, applications in diagnosing diseases and even treating cancer.
It sounds kind of backward, doesn't it? I mean, amino acids are the building blocks of life. But what if a cancer cell had a weird, specific dependency on just one of them? Well, if we could target that unique metabolic need, we can launch a very selective attack.
And that brings us right back to that enzyme, asparaginase. In the medical world, it's a powerful chemodrug used for a type of cancer called acute lymphoblastic leukemia. And why does it work? Because it turns out that while most of our healthy cells can just make their own asparagine, these specific cancer cells can't. They are totally dependent on snatching up asparagine that's already floating around in the blood.
So the strategy is simple but totally brilliant. You give the patient asparaginase, and it just mops up all the available asparagine in the bloodstream. Now, your healthy cells, they don't really care. They just make more for themselves. But those leukemic cells, which can't make their own, are literally starved of this essential nutrient, and they just can't grow anymore.
And the clinical connections don't stop with cancer. Remember AST, that enzyme that makes aspartate? Normally, it stays inside your cells. So if doctors see high levels of it in a blood test, it's a huge red flag that cells are getting damaged and leaking their contents, especially in the liver or the heart. Also, any genetic defects in these pathways can mess up the urea cycle, which can lead to a really dangerous buildup of ammonia in the blood.
Okay, let's zoom out one last time, because you really have to appreciate the elegant design here. Aspartate isn't just a donor, it's a critical bridge that connects several of the body's most important metabolic highways. This number, 1, represents something huge. It's that single critical nitrogen atom donated by aspartate that links two massive processes, the breakdown of proteins for energy and the safe disposal of their toxic nitrogen waste. It's one small atomic step, but it has massive consequences for our health.
And this is the masterstroke of metabolic engineering. Because aspartate is made from the TCA cycle's oxaloacetate, and then it donates nitrogen to the urea cycle, it creates this perfect, seamless connection between the two. This is no accident. It's a brilliant system that lets the body perfectly coordinate making energy with getting rid of waste.
Woo! We've covered a lot of ground. So let's just boil it all down to the most crucial, high-yield points that you absolutely need to remember.
If you remember nothing else from this explainer, remember this simple phrase: aspartate donates, asparagine carries. It perfectly captures their core jobs and will help you keep the rolls straight, no problem. Okay, quick check. Remember that key clinical point we talked about? That enzyme is asparaginase. It's that amazing tool in cancer therapy that starves certain cancer cells of the asparagine they desperately need to survive. And what about the core biochemistry? That whole transamination process, which is so central to all of this, is completely dependent on that coenzyme, pyridoxal phosphate, or PLP. And where does that come from? Vitamin B6. It's a great reminder of just how vital vitamins are in all these pathways.
And that brings us to one last big-picture thought. The success of asparaginase therapy is proof of concept. It shows the incredible potential of targeting these unique metabolic weaknesses in cancer cells. So as our understanding grows, what if we could find other asparaginate-like dependencies in other types of cancer? Could this be the key to a whole new generation of highly targeted, personalized therapies? It's a really exciting area of research, and it's a powerful reminder of why understanding these fundamental pathways is so incredibly important.